being recorded. Wow. All right. That's fantastic. All right. And interesting session as always. We're going to hear about use of mortality data and PPRL from Jasmin, Umberto, and Shaun. Monthly Recover Long COVID project team updates from Emily Pfaff and Melissa Haendel. And there is a N3C survey. And that is, I think, next in the thing, in the plan. So 2 minutes of your time, N3C user satisfaction survey. Like to hear from you as part of the N3C program evaluation. We're collecting information about your experience with N3C, utilization of N3C resources. Feedback is extremely valuable. And so I think we're going to take a couple minutes for people to do this, and the address is shown. And in theory, if you click on the link, it should take you there. I don't know actually if it will or not, but it's a short enough bit.ly address anyway.
Can somebody put the link in the chat, please? Yeah, Warren just wrote the same thing.
Okay, Melissa, why don't you tell us when we think enough time has gone by?
I don't know if Anne can tell if how many we've gotten responses, but I think we can go ahead and get started. And if people want to continue to fill the survey, we'd be very grateful. Maybe we'll post it again at the end.
But yeah, no, this would be a big favor. I think it'd be really great if people could do that, because this is very important to document people's feedback. Well, maybe it's very clever that it's counting up 2 minutes, but I think it's too late. I didn't realize that. Well, maybe. Okay. Anyway, so upcoming sessions. We have ML training, workforce and benchmarking, ethical AI by Susan Greguric next time, and the new N3C public health dashboard demo by Leonie and Dave. And then March 28th, Good Algorithmic Practice Initiative, Rationale and Overview, update from the Logic Liaison team, and update from the Education and Training Domain team. April 4th, data quality considerations for evaluating COVID-19 treatments using N3C, principles for accommodating missingness in their role, and analytic frameworks for EHR-based studies. And missingness is, as you guys know, is a really key and knotty issue. MLP team update, PASC clustering pipeline and gap insights with the authors listed. And then April 11th, we have the rare disease presentation network discussion, Melissa Haendel and Brian Laraway. Okay, so I guess we are now above 4.5 million COVID positive cases. Fortunately, case loads are decreasing, which is good. So now we're going to launch into mortality data and PPRL with Jasmin Phua, Umberto, and Shaun. And so if you guys could share your slides, I think that would be great. I'll stop sharing.
Certainly. So I'm going to kick it off. Can everyone see my slides?
Yep.
Perfect.
Good.
Great. This is the team that has been working on the PPRL component, but as you'll see from the as we kind of unfold this, there's numerous other people who have contributed and who have obviously been close partners. Just a quick orientation. I will be covering the overview on the mortality linkages. Umberto will be covering the linkage infrastructure, and Shaun will be doing the deep dive on matching methodology. And finally, we are going to work really hard to make sure that there's time for Q&A, because we know typically this session does engender quite a number of questions. So definitely hold me to my few minutes that I'm going to talk through, and we'll kind of pass it on. For those of you who participated last week, Shaun O'Neill provided an excellent deep dive into PPRL and what the mortality data set was. Please check that out if you missed last week's session. I'm going to provide the kind of Cliff Notes version of both this and the mortality data set before we get more into the processes. So for those of you who aren't familiar with privacy preserving record linkages, it's a cryptographic method of linking disparate data sets in a de-identified manner. So for example, in this illustration here, Jane Smith is seeking care at both Duke and UNC. In this case, both institutions would run data van software generating de-identified tokens. They would send it on to the N3C linkage honest broker, which is held by Regenstrief. And you'll see here these kind of like gobbledygook strings of characters. These are the tokens. Obviously, this is a kind of simplified version. It illustrates what one token looks like, but N3C actually has a design where there's a maximum of 18 of these that are used for linkage. The main concept here is when institutions send out these tokens, they're in a form that they can't be linkable. But Regenstrief, as the N3C linkage honest broker, is the authorized party who has the ability to process it. So what happens when the tokens for the Duke Jane Smith versus the UNC Jane Smith gets sent? It actually gets transformed by Regenstrief into the strings that you see here, where this is how the record linkage actually occurs. So the gobbledygook of strings that can't be matched turn out to be transformed into the same strings, and that's what PPRL is at its core in terms of the linkage. So feel free to pose questions later, or if you want to go in depth into this again, the training material in the Enclave has a step-by-step illustration of this that might help you understand it better. Wanted to just provide a high level of kind of three main use cases within N3C for which PPRL is used. So first is obviously deduplication in situations where it's care fragmented, where a patient may have records both in Duke and UNC. And then second, there are multimodal linkages, and Joel's presented on this in the past. So when we're linking to other repositories, such as the Cancer Imaging Archive for imaging data, or even viral variants that's coming up soon. These are the kinds of rich modalities that we want to be able to integrate within N3C. Finally, and the PPRL is also used for cohort discovery. So if all of us or Biodata Catalyst want to understand what proportion of their participants actually have data within N3C, they are able to generate tokens and kind of use it almost as a cohort discovery platform to determine if they're kind of data relevant to them that might be pullable. In all cases, it helps with study feasibility. Finally, in terms of PPRL-based data enrichment, and this is what we're going to focus on today, want to give everyone a sense of just the different components that we have running. So obviously, over time, more and more health system sites are generating their de-identified tokens. So that's on a rolling basis. Mortality data has been onboarded and is available in the Enclave. That's what we're going to discuss today. Viral variant data coming again from a series of health systems is being added on an ongoing basis. And we're very excited that we just kicked off the CMS claims linkage effort. So just wanted to give everyone a sense of like what's coming up. In terms of the kind of third-party real-world data, the purpose of this is to obviously have more complete and timely death reporting compared to what we're seeing in the Enclave right now. So what is being reported in the Enclave is kind of N3C health system sites, right? But what we do know is that is relatively incomplete, probably need to be enriched with others. And therefore, we have 3 real-world data sources. So 1 government data source, which is really the SSA death master file, and then 2 obituary data sources that I used. I won't go in too much detail into each of these, but wanted folks to just be aware that obviously there are other classes of data that hold mortality information that are being brought into the Enclave over time. You know, in the past, we've linked to NCI's digital pathology repository. Obviously, that is a gold standard because it's autopsies that were linked in. So, you know, I would encourage everyone to think of these in terms of different levels of authoritativeness, depending on where the provenance of the data is. Finally, in terms of the overall N3C mortality data flow, reading from left to right, obviously there are identifiable death reports in kind of third-party real-world data. That is then translated into tokenized mortality data. So they have a set of de-identified tokens that come with them. In N3C's unique process, the tokens go to Regenstrief as the N3C linkage honest broker, and the actual mortality data payloads, such as dates of death and dates of birth and that sort of information, go directly to the N3C Enclave. One thing I want to call out here is, you know, within N3C on the health system side, everyone's maximizing linkages by generating a maximum of 18 tokens, but the mortality data sets are much more restricted because they do not have all the underlying demographic elements needed to generate everything. So for the Gov data set, for example, tokens 5 and 16, which are SSN-based tokens, can be generated, but obviously you wouldn't find that sort of information in obituary data, and therefore the first 3 tokens are generated for obituary data. So as we kind of start thinking about linkage and data quality and matching quality, that's just something to keep in mind, as well as like what happens when investigators actually use this linked data set. On this part, we want to talk a little bit about the quality process, and this is the part that we are in right now. So the linkage honest broker, Regenstrief, obviously has all the tokens, and they are the token clearinghouse. The tokens are then linked and matched. When that occurs, they send over a linkage map to the Enclave. And what happens is the Enclave is going to generate a preliminary data set. There are a series of different data quality rules that need to be applied. We are currently in this stage of cleaning the data and looking for potential collisions. And, you know, again, the reason for that is, as you saw with the N3C sites, we know we have a max of the 18 tokens, but with the mortality data, you know, it's not equal. So we want to be very judicious with how we formulate linked data sets within the Enclave. And so there are kind of 3 main buckets that we look at. So are there demographic collisions? Did I link someone and one record says the person's 15 years old and the other record says they're 57 years old, right? The other thing is possibility. At the date of death that you get from the mortality data, do you see a ton of activities and procedures and medications after? Obviously something's wrong there, right? And then finally, we will be looking at provenance to create the final linked data set with the understanding that the gold standard where N3C EHR data always precedes everything in terms of the authoritative data set. So that's the kind of overall process that we have within the Enclave right now. And I just wanted to have a call out to the Palantir team and the data ingest and harmonization team. Everyone has been fantastic, working very, very collaboratively and iteratively through this process. So we can kind of make the final linked data set available to the community. I'm going to pass it now to Umberto.
Thank you, Jasmin. Let's see. So I'm gonna briefly present the architecture of our platform, the Linkage Jonas Broker. You see here on the left. Let's see if I can have some. On the top left, you can see that the institutions, they process the files and they produce the pseudo identifiers or the tokens using the PPRL, using the PPRL software that Datavant developed and was presented by Jasmin. And then we receive those tokens. We store, organize them, and we also, when we do that, we also start the pre-matching process, so we can start to identify duplicates, for instance, which is the most common use case. But we still keep all the tokens there for other linkage needs. You can see on the bottom left that the Enclave receives the data. So we receive the tokens, they receive the data. And when we need to provide linkage maps back to them, this happens on the arrows to the bottom right. And the authentication of users, etc., is planned to rely on the RabbitMQ service operated by NIH. So I know it's a loaded slide, but this one is probably a simpler version of that. So we also produce the linkage for data that is external to the N3C Enclave. And those are the use cases listed to the right. Images, and Jasmin went through those before. CMS claims, mortality, viral variants. We are very close now to start getting the CMS data. And I saw that there was a comment that it's very exciting that we will have the claims. But also mortality, I think it's very critical, and that's the main focus right now. So what happens, what is different here is that instead of producing maps of linkage that goes to the Enclave, in this case, we produce linkage maps that goes sometimes to the Enclave, but sometimes to other infrastructure, for instance, Midric for images. So the onboarding process, just to go quickly, prerequisite is to have the Linkage Jonas Broker agreement signed. Step one is site completes registration form, whitelist IP address, and then an SFTP account will be created. Individual accounts will be created, and the passwords will be granted to the users of the sites. And then users connect to the SFTP when they are sending us the tokens. And then finally, the data is transferred to Linkage Jonas Broker. The infrastructure behind the SFTP is FISMA moderate. So it's a highly secure environment. The use cases are here, just so you know, just so you have a list again. Mortality, all of us, biodata catalyst, TCIA, Midric, CMS, and viral variant. So those here are the current matched numbers. And you need to take that, understand that, I don't know what, the numbers are still, the methodology is still in fine-tune mode right now, but those are the match that we have among the pairs, the use case pairs. N3C versus N3C, N3C versus all of us, Biodata Catalyst, and Biodata Catalyst and all of us, and Mortality on the bottom. So you can see that it's quite an important number of matches. Here is just so you understand for mortality more specifically, we are using 3 data sources, SSA, Private Orbit, and orbit.com. And then we consider we consider mortality using a logic of having more than one or more confirmations from one of those feeds, but it's important to understand that we also provide the individual matches back to the Enclave. So if someone wants to understand where the mortality information is coming from, that's also possible. And let's see where I am. So I'm gonna pass now the mic to Shaun Grannis and try to control the slides.
All right. All right, thank you, Umberto. We'll start at matching approach. One slide backward. Great, thank you. I don't know if people can see me or not, but I'll just go ahead and talk. So in order to develop a PPRL algorithm that exhibited ideal match characteristics, which are, of course, high precision, high recall, and what we did was we evaluated an initial match algorithm using gold standard data generated from the Indiana Health Information Exchange, which contains over 100 different health system sources. And so using that data, we initially evaluated and identified 18 token pairs, token sets that we would incorporate into a deterministic OR algorithm. Once we deployed that and began evaluating, we reduced the number. Could you, next slide, Umberto? We revised the tokens to a set of 14 that are in gray here. Those not grayed were tokens under consideration initially, but then the gray tokens are where we have landed. Right now, we are running a deterministic OR algorithm, as we call it. And what that means is if any of these tokens match, we declare the records to be a match. We'll continue to evaluate the data quality, the match quality, the cluster sizes, and make revisions as we move forward. So we wanted to show you some basic statistics from the type of data that we are receiving right now. So what you see here, if you could go to the next slide, Umberto. What you see here are just the percentage of total possible tokens generated that are received by the Institute. So for example, I would just, let's use the top site as an example, Washington University, St. Louis. They are sending us a total of 4.6 million tokens. There are 18 tokens per record. And there's a total of 258,000 token sets or records that we've received. Of those tokens, 31% or 1.4 million are missing or not present. So we monitor the completeness of the tokens to understand the quality of data and will provide feedback to sites for particular tokens. again, this is one of our ways of monitoring the algorithm, and of course, data quality is an important contributor to overall match accuracy. Why don't we move on to the next table? And here are broken down by individual tokens. We can actually evaluate and look at particular issues. So across the bottom of the screen, you see the average completeness for each of the tokens we are currently using. And so token 3, which is last name, first name, date of birth, and zip 3, that is relatively complete at 97% overall. Many sites are sending 100%. But then if you look at, for example, token 39, which is SSN and date of birth. So any organizations that do not have SSN or are not sharing full SSN would not be able to generate that token. So we're seeing about 40% completeness for those tokens. And again, we use this on a regular basis to evaluate the token completeness to identify any issues with those tokens. In addition to monitoring completeness, we can also look at token size, or excuse me, cluster size. So we can look at how many different sites are being linked by a particular token to identify any overmatching or overly sensitive or, algorithms. And in monitoring clusters and monitoring these performance metrics, we're able to evaluate and refine our algorithm. So with that, I would like to end the presentation, my part of the presentation, and would welcome any questions now or later. Thank you.
So just a quick comment before we continue here. So when Shaun mentioned social security numbers not being available, none of the identifiers ever come to the, I want to emphasize that. So what happens is that some places have their identifiers, including social security numbers. If they do, those identifiers will be used to create the tokens, but the numbers themselves will never be known, and they cannot be, and the tokens cannot be reverted back to any true identifier, just to be completely clear here.
Yeah, thank you, Umberto.
Great. Well, this is fantastic. There's been some great discussion in the chat, and obviously tremendous amount of enthusiasm, including from me, about the implications of this. It's incredibly exciting to see this come to fruition and be something that will really actually be useful. And viral variant CMS linkage are, incredibly would be incredibly impactful. I think one interesting point that was in the chat, just from a logistical point of view, it seems to me that many or most DURs will end up being revised to incorporate this. I mean, pretty much every research project, it's hard to imagine you wouldn't want CMS data, you wouldn't want death data, you wouldn't want viral variant data. So that's something that I think Ken had mentioned. There's obviously be more discussion on how to do this efficiently.
So Joe, can I ask a question, and maybe just for our action items? you know, I think you're absolutely right. This is like so exciting. Everyone's going to want it, and everyone's going to want it at the same time. And so the DAC is going to be entirely overwhelmed by requests to update DURs. So I just think we should be a little thoughtful about how we suggest to folks to kind of time the rollout of the updates to the DOR so we don't overwhelm the whole system. So just wanted to make a note of that for our future selves.
That's a great point, Melissa.
Great. Yeah, no, that's fantastic. A question about the pathology data. I suppose I'm sort of abusing my role as moderator, but that's sort of interesting. So how widespread is this, and where is it coming from?
Are you done? Sorry.
Go ahead, Ken.
Sorry, Jasmin.
Anybody who knows the answer, I was intrigued to see that that was one of the things being linked.
I think, Jasmin, you're referring to the digital path from Janelle Kortner.
Yeah.
So when we were testing the system, we did pilots with, obviously TCIA was one of them, and a digital path was one of them. And these were pathology reports from the state of Maryland on people who had passed away from COVID.
Okay. Okay, great. Okay, well, this is fantastic. I think we should move on to our next thing. This is fantastic. We could continue for a long time, but I want to make sure there's time for the Recover Long COVID project team updates. So I hand it over to Emily and Melissa.
Okay, I'm gonna do the slideshow version here. Let me know if you can see it in presentation mode now. I can't see you anymore, so.
Yeah.
Okay, great. Okay, and these slides are posted at that bit.ly. We can put the link in the chat. So this is starting to be a regular occurrence. Once a month, we are trying to give an update on our very large long COVID EHR Recover project. And so we're trying a little bit of a new format today about some of our thematic areas. And so what we had planned to do in future iterations of this presentation and update, and it may be other members of the Recover EHR group that present in the future, is to just try to give you the general categories of work that we're giving you updates about. Much of the work is in process and has not been published or made publicly available to date, although there are many resources that have been created that are available within the Enclave or on GitHub. So, but this is just kind of a true where we're at right now update. Let's see. Oops. So I wanted to remind everybody, just at least on this round, who the principal investigators are and who our operations folks are on this slide, in case any of you have questions, especially sites who are actively participating in the EHR Recover program. These are your go-to people to ask questions and figure things out. We're always happy also to help with the various NIH engagements that are necessary to make all of this go. So, and again, these slides will be made available. I also wanted to thank all of the sort of deeply contributing sites. We funded as many sites as possible to participate in this effort. And so I wanted to thank those who have contributed data, analytics, expertise, and more. So as you all know, long COVID phenotyping has very many challenges. It's, as a new disease, we see a heterogeneity of signs and symptoms. Some of these are clinically assessed, some are self-reported, and we really need a much more holistic picture of the patient and their longitudinal trajectory, as well as an understanding of key factors such as the timestamp of a diagnosis, a positive diagnosis for COVID, or vaccination status, and other sort of comorbidities and their temporal evolution as well. And so that sort of how those features relate to the longitudinal and episodic nature of how we evaluate symptoms is also a challenge. We also have a pretty extraordinary variation in care-seeking behavior across different age groups, gender, different communities, and with different social determinants of health attributes, and urban versus rural. And not everybody has access to a long COVID clinic, nor does everybody get a second visit at a long COVID clinic. And the clinics themselves have different practices for which patients they are willing or able to see. And then we also have a lot of variation in the timing and nature of COVID testing, and many, many, many long COVID patients that have never been tested tested positive for COVID. So this is the sort of landscape in which we are trying to do our analytics. No small challenge, but we have some great work underway. And so these are the categories that I mentioned before. You've heard us present earlier on some of our earlier phenotyping work and defining PASC, and we're going to give you an update today on our multiple classification strategies and how we're characterizing our cohort. We have some new work going on clustering and phenotyping, subphenotyping, really trying to understand what subtypes of PASC actually exist. And some different strategies for revealing those such that they might be targets of different types of interventions or risk prediction, which brings me to the next one. What care is being given now in terms of interventions, and what are the key risk factors that might be potentially ameliorated if we can identify them? We've been working with the Patient-Led Research Consortium, the PLRC, in particular, to work together on understanding how to best include patient-generated data in our evaluations. We recognize that that is a core need, because the patients report different data than we have available to us within the EHR. And then also making sure that we're answering key questions that long COVID patients have in understanding their condition and their trajectories. And then finally, a very brief update on some data enhancements that sites are making to support the above areas of focus. So this is the sort of format that we are hoping to present in the future in terms of progress in these areas. If you have any feedback on this or interest in areas that you know we're working on or think we should be working on that are not in one of these buckets, please let us know, and we'll just keep iterating. So I'm going to hand it off to Emily, and if you just want to let me know when to advance the slide.
I will do. So I wanted to do a little bit of a deep dive, a shallow deep dive on our long COVID phenotype as it stands today, because I realized that I've talked about our phenotype a lot on this venue and other venues, but I haven't really gone into detail about what it means to phenotype, do computable phenotyping using machine learning. And so I thought that it might be good to sort of back up a little bit and do an explanation of what does it mean when we say we have a machine learning based phenotype for long COVID. So the first way that I will explain it is by saying what this is not, which is a rule-based computable phenotype. I've been computable phenotyping for my whole career, and generally when I do computable phenotypes as part of research studies, I make rule-based phenotypes. I have an example of one here where we're defining a patient with diabetes with a patient with a certain number of ICD-10 codes, and then maybe a prescription for a diabetes drug or a max HbA1c of 7% or more. Those are very clear rules, but one investigator may disagree that those are the right rules from another investigator, but at the very least, we can say that they are straightforward and able to be interpreted by anyone who looks at the definition. But when a disease is new, like long COVID, there aren't those clear and straightforward rules to codify. There's nothing that I can translate into code as easily as the example that you see above this. And so when faced with this, we opted for another phenotyping method, which is a machine learning based method. Now, the machine learning based phenotypes are not always the most appropriate way to go. And in fact, I usually think that the simpler approach is better. But because that was not possible here, because we have enough data, certainly within N3C, and because we were able to identify a set of patients that could work as a training set, we felt like machine learning was a really good alternative here. Next slide. So when we train a machine learning model in the phenotyping world, the first piece of the puzzle is that we need some group of patients that meet our case definition. And we need to feel pretty good about saying, these are the true positive cases. Now that was particularly complicated with long COVID because even that is difficult at this point. It is difficult to say this patient definitely has long COVID and this patient definitely does not. I would venture to say that we can't do that today, and we couldn't do that when we started this work. So what we needed to do was come up with what we've been calling a silver standard, which is not quite as good as a gold standard, but it is the best available proxy that we could come up with. In the case of our earliest model, the proxy that we came up with was anyone who had a visit to a long COVID specialty clinic at the health system where they get their care. Now, do 100% of those patients have long COVID for sure? Probably not. Is that population highly enriched with people who do have long COVID? Yes, at least that's our hypothesis. And so those became our patients who met our case definition. And our case definition actually wasn't patients who have long COVID. It was patients who sought care in a long COVID specialty clinic. That way we know that those patients truly do meet our case definition. But then in the rest of the enclave, we have tons more patients, millions more patients who might meet the definition and might not. We don't know anything about them. And that's what we need to build our model to find out. Next slide. So let's just look at our training patients for a second. These are the patients that we feel good about, calling them true positives. Now, I have sort of made up this list based on some of the stuff that we saw. So this isn't like a real list of features that all of these patients have. But this example is to show that a machine learning model looks at all of the patients that we've defined as being our true positives. And we give that model all of the available data about those patients formatted in an organized way. And that enables the machine to basically understand patterns among those patients. So you see here that I'm saying that demographically, these patients are very different. So I'm the machine in this case, and I can't make any assumptions or patterns based on their demographics. They're all over the place with age, race, ethnicity. All right, so that's not that useful. But I am seeing that many of these people have a new prescription for an inhaler that they didn't have before they had COVID. Same with antidepressants. Some of them, not many of them, but some of them, enough to be interesting, have a prescription for a sleep aid. And again, some, not all, not many, but some are in physical or occupational therapy. Seeing a lot of respiratory complaints, and fewer, but still significant number with gastrointestinal complaints. And when I take a look at their lab results, I'm not really seeing any signal there, not terribly similar. So at this point, the machine gathers up all of this information that it has, where it knows that there are some things that are present in lots of people, and those become important. And then there are some things that are present in more people than you might think, and those are also important, although maybe less so. And then there's stuff that just looks like noise. And that stuff either falls to the bottom or just doesn't get used at all in the model. And of course, because there are hundreds, if not thousands of variables, in fact, there are thousands of variables in our model, this is way more pattern finding than a human could do. You need a machine to do this. Next slide. So what we end up doing then is taking the model that's built off of our true positive patients in blue, and we run it against the rest of the population. When we do that, what we end up getting out is not a, yes, this patient has long COVID and this patient does not, but rather we get a predicted probability. And so the model, it's a little uncertain. It sort of hedges its bets. It doesn't want to say yes or no for sure, but you can see that the patients that I have circled in orange here, that I'm calling patients who look like the training set, are patients where the machine is highly confident that they really look like the people in the training set and are highly likely to be true cases. And then you can see on the right, people that I have not captured are people that have low predicted probabilities. And so those are people who don't look like the training set, and we are prepared to call them, again, as proxies, not long COVID. Next slide. So right now, as of today, we have 3 different training sets that we use to train 3 different models. And we do this because we are experimenting. This is a very new area of research. We don't want to settle on our first model. We're trying a bunch of things. Just to give you a little inventory of what we have, our first model that is currently available today is, as I mentioned, based on patients who visited the long COVID specialty clinic. The training data for that model came from 4 sites out of all of the sites that contribute to N3C, but more are coming soon. Our second model, patients that have a U09.9 diagnosis code. That is the ICD-10 diagnosis code for long COVID. You may say, why didn't you use that code from the very beginning to define your cohort? It's because it did not exist until October 1st of 2021, which was just a few months ago. So we want to make sure to include patients that were in our system far before October 1st of 2021. But if you have a diagnosis code, we're certainly going to find that interesting and use that as a model. Right now, we're getting UO9.9s from 29 sites, and that's going up over time. And then a third model where we combine the training sets from those prior two models and then retrain the model on the combined set. But all of these models capture a slightly different group of patients due to being trained on different data. And you can get the data for number 1 now, and number 2 and number 3 is going to be available by this time next week. Next slide. So just, I put this in here just to show you that we feel pretty good about the performance of our model. So you can make a model for anything, but it doesn't necessarily perform well. If it performs at, 50%, that's no better than a coin flip. Our models perform reasonably well, and in some cases very well, when trying to identify patients who were hospitalized with COVID, not hospitalized with COVID, and then everybody together. We think we can even further improve on this performance, but this is the performance for model number 1. Next slide. And I'm not going to go through this today in the interest of time, but I do want to just put a plug in for explainable machine learning, because I can tell you all day long what the performance of my model was and that you should just trust me. But instead of just trusting me or trusting the machine, we think it's really important to be able to show our users and other researchers the different features that the model sort of hung on to for making its predictions. And so we can output plots like this that actually show what features were most important in making the decisions to see if they make some good clinical sense. And that's really, really important. Next slide. So I just want to summarize by showing that we have these 3 models. And then if you are looking for a link to the actual model output and you are on the long COVID DUR, that link on this slide, which we will share, gets you right there. And we also have a publication in press about that model that we are very much looking forward to having out. We will do the same for the other 2 models that you see here, and we will continue to fill in the blanks on this table to make sure that folks have access to this, again, as long as you're on the DUR. Next slide. Okay, so I'm going to either hand it off to Melissa or Richard to talk about what we have done with the output of those models to make sure that not only are we identifying patients that are probable long COVID cases, but we're also able to do analysis on that cohort. So Melissa?
Yeah, I didn't see Richard earlier and I can't see now, but if Richard, if you're here, feel free to pop in. Otherwise, I'm going to put my Richard hat on. Okay, so this is actually really nice, and we prepared this for a congressional update that the NIH had to give on the RECOVER program in order to help Congress really understand the potential impact of long COVID on our population in the different states. And so one of the nice things that we can do with the outcome of that first model that Emily so nicely described is really kind of look at the incidence and geographic distribution at different thresholds of probability. So if you recall, the populations that are identified at a certain percent on Emily's slide, that's a variable cutoff that we can use in the model. And so when we look at a 50% threshold over on the left, that's a much more inclusive threshold for defining that long COVID phenotype using the machine learning model. And you can see the percent of the number of patients that are affected in each individual state with the numbers indicated by the size of the yellow circle. And then over on the right, you can see what that looks like when we have a much stricter classifier used to classify those patients in the different states in which regions are most affected. Again, if these are, if you're on the long COVID DUR, you can get to the code. And we have some information on the dashboard. We are working on having more of this information displayed on our regular public dashboard as well. And so that's an ongoing project. And if you have any insight about what you'd like to see, please do let us know.
So, excuse me. So somebody promote Richard. Oh, sorry, Melissa.
Go ahead.
Go ahead.
I couldn't hear what you said, so.
Oh, Richard
He
is
was asking
here.
for someone to promote Richard. He's not a panelist.
I see. Okay. Yeah, and Richard might like to. Did somebody get Richard up? We're going to run out of time, so.
Hello.
Hi. Do you want to do this one?
Sure.
Okay.
So what we're looking at is the index date of their original COVID infection and the number of patients with that index date that would go on to meet the definition of PASC based on the computable phenotype that Emily talked about earlier. So it's really the relative fraction of people that will get PASC. So it's like hopefully perhaps related to the conditions on the ground at the time, maybe the viral variant, which is why we have the approximate error of the major variants on the bottom in those three primary colored bars, showing where alpha was dominant, delta was dominant, and now Omicron. And you might notice that the data don't overlap with Omicron. It's because we can't really make predictions about who will develop PASC until a sufficient amount of time has passed since the index COVID infection.
So stay on because we'll come back to you, Richard, if I can rush through this part. So we have a new clustering and subphenotyping group, and they're going to report out in more detail in a few weeks. And so I'm just going to give a quick overview of some of the different strategies that we're using. And this group is led by Justin Reese and Charisse Madlock, and then Shaun O'Neill is leading the topic modeling group. So the semantic clustering is based upon the human phenotype ontology. And we have some new functionality in the Enclave, and this is relevant to all domain teams, not just the long COVID one. It's also making my heart sing because it's really the translational science dreams that I'll get to in a minute about being able to sort of leverage more robust ontologies in the context of our clinical data alongside mechanistic data. So those packages include the LOINC to HPO package, the OMOP to OBO mappings, and some algorithms for semantic similarity that measure patient-to-patient similarity. We also have some topic modeling approaches, which really comes from document analysis, which essentially patients are like documents, and how do we find words that are in common using OMOP concepts, and essentially interrogating movement between topics pre and post-COVID. Our preliminary results have identified potential post-acute trajectories and risk factors using this type of approach. It's quite a novel approach to apply this sort of almost information science approach to EHR data in this context. So we're looking forward to hearing more results on that soon. And then similar, more standard machine learning methods like Emily described, looking at clustering based on subphenotyping based on meds, labs, diagnoses, temporal progression, and other facets. So just to kind of walk you through some of the really exciting results to date. And again, this is just work in progress. So for the OMOP to OBO HPO mappings, this is essentially taking sets of OMOP codes and annotating them to individual human phenotype ontology terms, which have more sophisticated semantic algorithm capacity, as well as data linkage to basic research data. In this context, we take measurements, conditions, drugs, immunizations from the condition occurrence table, as well as the creation of long COVID code sets. And this was work that Tiffany and Peter Robinson and I and others had done to sort of say, what are the long COVID features that patients have identified, and creating code sets from OMOP and mapping those to the human phenotype ontology. So that's all available there in the Enclave. We also have the LOINC to HPO, which essentially takes laboratory measurements and their values and maps those to the human phenotype ontology. So you can have like abnormally high blood glucose, for example. And these come from the measurement table. And these are also deployed in the N3C Enclave. Now, once we have these things in the Enclave, now we can actually leverage some of those semantic similarity algorithms that depend on a robust ontological framework. And this includes things like Mbigin and Nsmallin, which are algorithms for graph machine learning, as well as mechanisms that we can use to use the COVID KG, which includes a whole bunch of different kinds of basic research resources that have been integrated into a giant knowledge graph, and that's led by Justin Rees. And then a heterogeneous version of Node2Vec, which sort of also helps sort of evaluate these large graphs. And you can see an example of what those clustering patient-to-patient similarities would look like when, and this is not showing any unapproved data downloads, don't worry. But you can kind of see what we're aiming for in terms of the output of having put all of these different pieces together. And then again, because we have these the knowledge graph with the mechanistic data in there, we can start to look at correlations between mechanistic underpinnings for each one of those clusters, which will give us key interventional targets and key risk factors. And so this is kind of what that looks like. So you can see over there on the left, we have a long COVID patient with some different findings. Over here on the right, we're trying to match patient-to-patient similarity. This patient has 2 terms in common, but some terms that are related but not exact matches. So acute kidney injury is related to transient renal failure. And using the ontology, we can determine that these 2 things have a relatedness. Same thing with the cough and the respiratory finding. And that's where the use of a semantic algorithm can really get us some new approaches to clustering with EHR data. So what we do is we calculate the information content of each common ancestor for each pair of terms. So we want to know, for example, that acute kidney injury, how similar is that to reversible renal failure? And using those maps from OMOP to HPO that Tiffany Callahan generated, we can create a patient-to-patient set matrix of similarity and then use clustering algorithm to create those clusters. And so this is what that looks like when that gets output. So now we're going to go on to interventions and risk factors. Would you like to run through this real quick, Richard? We have 1 minute. Maybe we should just end here and save it for the next time.
Sounds good.
Okay. So yeah, we'll have to maybe schedule a longer update on the next round, or maybe there's not enough time for each of the buckets every time. I think we ran out of time. Sorry about that, everybody. But hopefully that was helpful in terms of understanding what the long COVID group is doing. And again, a lot of these methods and artifacts that are being created and linked together in various kinds of ways for the purposes of the RECOVER program are also critically important for other areas of COVID research and the other domain teams. And we're trying to test them as best we can and get them out to the rest of the community as quickly as we can. Please do let us know if you have any questions or interests or think that there's other pieces of those puzzles that we should be considering.
Yeah, no, I absolutely think that it would make sense to have a longer presentation and discussion, because clearly these methods are very broadly applicable. So maybe we can go offline and find some more time during one of these sessions. So
thanks everybody.
Fantastic. So we're out of time. I apologize for not being able to tackle any questions, but we'll revisit this very soon. Thanks everybody. Take care.
Bye. Thanks, Joel.
Bye.